My protein is piezo1. And then the mutation is arginine 2456 is being replaced by histidine R2456 H. The normal function of my protein and the type of function that the protein holds. The normal function of the piezo1 protein is that it acts as a mechanosensitive cation channel, sensing mechanical forces and transducing them into electrical signals, which then alter cell activity. The protein functions as an ion channel, and it usually detects things like external signals and translate those signals into intracellular responses. Piezo1 doesn't alter chemical reactions or regulate genes directly. Instead, it acts as a reciprocator that activates downstream cellular signaling responses in response to mechanical forces. The protein is normally found in the plasma membrane. Red blood cells, smooth muscle cells, and cartilage cells are just a few that contain piezo1. This protein is mostly found in animals, but can also appear in plants because some of them have mechanosensitive ion channels. The type of change that the type of change the mutation has on the protein, it makes the protein less active. It makes the protein more active, and it actually makes the protein overreactive. The main functional groups present in the original side chain are arginine has a guaninidium group in its side chain, and the group is positively charged and consists of 3 nitrogen atoms bound to a central carbon. It is considered polar because the guanidium group is positively charged. As I said, it's positively charged. And how would I characterize its size compared to the other 19 standard AAs? I would consider it large because of its long, complex side chain. The interactions that originally can participate. Okay, so. Okay. Arginine is good at making connections with other parts of the protein or nearby molecules. It has a strong positive charge, so it's naturally drawn to negatively charged amino acids like guanylate or aspartate. The bonding of these helps start forming ionic bonds. Its side chain also has nitrogen atoms that can form hydrogen bonds. Excuse me, which help hold the protein together. Plus, if it interacts well with polar areas, parts of the cell that have phosphate or carboxyl groups, or anywhere water is present. The main functional groups present in the mutated side chain. The mutated side chain was the histidine contains the imidazole ring, which is a 5-member aromatic heterocycle. This means it has a ring-like structure containing 3 carbon atoms and 2 nitrogen atoms at positions of 1 and 3. They're not adjacent to each other, and this can also act as a weak base. It's hydrophilic. It's neutral. It's neutrally charged. It's hydrophilic, and how would I characterize its size compared to the other 19? I would say it's around the middle of being small or large. It's definitely smaller than arginine, but it is not too. But it is not as small as something like Eileen. The differences in the mutated AA is that the histidine can act as a proton donor. This allows them to function as an acid and a base, simultaneously providing both hydrogen and hydroxyl groups. Histidine can still form hydrogen bonds, especially using the nitrogen atoms in its imidazole ring, but it doesn't hold a strong, consistent positive charge like arginine does. This means it's less reliable when it comes to forming ionic bonds, and overall, the electrostatic interactions just make it weaker or just different. Another key difference is the size. Histidine is noticeably smaller than arginine, which can impact how tightly things are packed in the protein and might affect how stable that part of the structure is. Histidine is also more chemically flexible. It's like I said, it's imidazole ring can gain or lose a proton depending on the local environment, making it more reactive and better suited for roles that involve transferring protons. The assigned mutation affects the my protein function because R2 456H is in a critical place for channel gating and the ion conductance. Replacing arginine with histidine can reduce the positive charge. This would affect the electrostatic interactions, which regulate the opening and closing of the channel, which can cause a lot of problems if it doesn't function properly. It can also lead to an abnormal ion flux, which can disrupt cellular signaling. The mutation is associated with xerostasis, which is a type of anemia that can be hereditary. This happens due to red blood cells being dehydrated due to the increased ion efflux. Now, the overall shape of my 3D protein is about like a hoop with 3 prongs stretching outwards, and then the additional subunits, as you can see. Like I just said, the additional subunits you can see, I represent them by showing the different colors of my designs and the different pieces. As you can see right here. My assigned mutation doesn't truly affect the structure much, other than making it a bit smaller. And yeah. The assigned mutation affects the protein's function by making the protein more active because it makes the protein overreactive. The mutated amino acid is it is in the C-terminal in the cap domain, which is shown here in my model. And then the limitations for the model was that not everything could be shown that I wish it could have been, especially because I had used pipe cleaners and could not add too much detail. But still, I feel like that comes with every model, except for unless you had like actual design you could have in a computer or something of the sort. And yeah, thank you so much.